Goodness me, what a turn up. You should give yourself an applause. This is fantastic. I mean, you're even behind me. My name is Natasha Mitchell from ABC Radio National. I'm a science journalist and presenter of a program called All in the Mind on the network. Delighted to be your host this evening. This event is brought to you by Melbourne Conversations, the City of Melbourne's free public program of talks, debates, conversations, and also by Future Leaders, another initiative. I thought I'd put on the public record that I think Woody Allen was wrong. He was wrong when he said that his brain is his second most favourite organ. I'm prepared to shamelessly admit that the brain is my favourite organ. And tonight we're in for an enticingly optimistic and hopeful take on this brilliant organ of ours. And as you'll hear from our three speakers, our brains have in fact underestimated themselves. Few bits of housekeeping. Turn your bits of technology off for us. I'd be most grateful. There'll be plenty of time for Q&A tonight. We really want to get your questions and input to the discussion as well. And I'm sure many of you have seen Norman Doidge on the 7:30 report, so you've probably got some questions to pick up from that as well. Also, a reminder, we're filming today for possible broadcast. And another thing, you're probably sitting on it, there's a survey. We'd love you to fill it out. The City of Melbourne really values your input and wants to constantly build and grow this program. And it's also a way of actually getting your name on the mailing list so you can get emails or mailing lists, whatever format, usually email, to hear about future events in the series. Let me introduce our international guest tonight. Dr. Norman Doidge is a research psychiatrist and psychoanalyst on the faculty at the Columbia University Centre for Psychoanalytic Training and Research in New York and the University of Toronto's Department of Psychiatry. He's also a multi-award winning writer, essayist and poet, a true polymath. I think he won his first poetry prize at age 19. Very impressive. His new book, which is on sale at the back, The Brain That Changes Itself, Stories of Personal Triumph from the Frontiers of Brain Science, really has caused quite a storm, especially in the USA when it first came out. I gather it's been on the New York bestsellers list, and that's always an author's dream. And I've read it, and I can testify that he really has pulled together some utterly compelling stories about the capacity of all our brains, even those that have been irreversibly, seemingly, damaged by stroke, genetics or disease. All our brains have the capacity to thrive and evolve, and I want to give Norman Doidge a warm welcome, a warm Melbourne welcome. Let it rip.
Thank you very much. It's really a wonderful honor to be here at this civic conversation to talk tonight about the so-called plastic brain. The idea that the brain is plastic in the sense of changeable, adaptable, malleable is, I've come to believe, the single most important change in our understanding of the human brain in 400 years. It's revolutionary. And since all human activities emerge from the brain, any change in our understanding of the brain ultimately has a major impact on anything that we do. I define neuroplasticity as that property of the brain that allows it to change its structure and its function. And that's in response to the actions that we commit ourselves to. It's in response to sensing and perceiving the world, and even, quite fantastically, to thinking and imagining. Some very, very brilliant experiments conducted by Dr. Eric Kandel at the end of the last century ultimately led up to the demonstration that human thoughts and learning actually turn on certain genes in our nerve cells, which allow those cells to make new connections between them. Eric Kandel was a psychiatrist who actually wanted to be a psychoanalyst. He came from Vienna, and when you think of that, you think of Freud. And interestingly enough, it was Sigmund Freud who in the 1880s and 90s first speculated that thought actually leads to changes between the connections in the brain cells. And in fact, he called it the law of association by simultaneity. Now, neuroscientists say neurons that fire together wire together. Freud called it the law of association by simultaneity because he thought when you experience two things simultaneously, the brain wires them together. So that's in part why you have a psychoanalyst here talking about brain plasticity. Another reason is that when I would be working as a psychiatrist and analyst with patients and they didn't get better, my colleagues were increasingly saying, well, these problems have been hardwired into their brains. Because I was a poet, I could tell a metaphor when I heard one, and I knew it was a metaphor because, yes, when the same patients did get better, nobody mentioned anything about hardwiring. Hardwiring was a kind of shorthand for the notion that these circuits had been genetically predetermined. This whole area has the sense of the fantastic about it, the idea of thought changing the structure of matter. The Western tradition begins with the notion that thought changes matter in the sense that the Old Testament begins and God said, "Let there be light," and there was light, and words led to the change in the structure or the development of the universe in that story. And of course, the New Testament says in the beginning was the Word. And Plato spoke about the superordinate power of ideas and forms in somehow or other governing our lives. But these were in some respects supernatural ideas, the idea of thought changing structure. The Bible doesn't have the word nature in it, actually. These were extraordinary events, and yet I can just will to raise my right hand, and my right hand goes up. And in some ways, it's a very ordinary event. So we're in some ways very much of two minds about this whole notion of thought-changing structure. Now that we're beginning to understand how these structures change, brain plasticity has implications for medicine, psychiatry, psychology, human nature, love, sexual attraction, acculturation, acquired tastes, our understanding of why addictions happen, pornography addictions. They have implications for politics, for business. Anything having to do with human training or education has to be re-examined in light of neuroplasticity. In fact, anything having to do with culture has to be re-examined in light of neuroplasticity, because it's no longer sufficient to say that the relationship between the brain and culture is a simple one where the brain produces culture. We now know, in fact, that culture also rewires our brains. Neuroplasticity gives incredible hope, and I'll give you just one story. But it also tells us that this resilient brain that's emerging is also a more vulnerable brain. It's very sensitive to what other brains do to it. And we're very influenced by our technologies. So when I first got the sense that the human brain might be plastic, it seemed that there were these people working, scientists and of all kinds and clinicians, who were working in very different intellectual silos, and they weren't always talking to one another. And I set out to find people, clinicians, at the cutting edge of this astonishing science who had already used it to transform people's brains. And so that I could actually see with my own eyes what was happening. And if I heard that someone allowed someone with a stroke to get better, I wanted to be able to push against that person's hand and feel how it had changed. And in the course of my travels, I met a woman with half a brain that had wired itself. I found many instances of learning disorders that were completely cured. Blind people who had been congenitally blind since birth, learning to see in new methods. Strokes and brain traumas changed, chronic pain erased. I saw children with cerebral palsy, a severe condition, learning to move their little hands more gracefully. And in my own practice, increasingly saw examples of how these principles could be used to alleviate entrenched anxieties and depressions. Now, the reason that this is a revolution is because for 400 years, our best and brightest scientists actually thought of the human brain as a complex machine, like a computer. A machine with parts, with each part performing just one function. And that is very much alive and with us today in the notion of the brain as a computer, or every time you open your newspaper and you see a picture of a brain scan and they say, this is where your brain processes music, in this part here. Well, it's all much more complicated than that. But this notion of the brain as a computer gave rise to what I've called a neurological nihilism, because it meant that those who were born with brain deficits or brain limitations had necessarily to live with them, in all cases. It meant those who had sustained brain trauma, in all cases, could do nothing about it, because machines do many glorious things, but they don't grow new parts, they don't rewire themselves. In medical school, I was taught all of these things, and that there were no new cells in the brain. And anyone who had a normal brain, who hoped to improve it or maintain it as they aged, was probably wasting their time. And above all, human nature, which was seen to emerge from the brain, was also seen as necessarily fixed insofar as we saw the brain to be fixed. Now, all this turns out to be spectacularly wrong. I'm not saying that the brain is infinitely malleable. I'm not greeting a bad metaphor with another extreme statement. I'm saying that we've underestimated what our brains can do, and that we have to move more to the sensible middle in understanding and appreciating what change is possible. And the following question, of course, must occur to each of us, which is, okay, so if the brain is plastic, if it's been plastic since the beginning of our species, how is it possible we missed it? And there are really 4 reasons. The first one is a very simple one I'll dispense with quickly. It's we didn't have the technology to view it. We needed to be able to make microscopic movies of the brain, because the brain change takes place microscopically. The second reason is a profoundly important one, and it's the mechanistic bias. The ancient Greeks didn't have a mechanistic view of the brain or nature. They saw all nature as one vast living organism. The microcosm, the macrocosm in some ways reflected each other. And anything insofar as it moved was thought to be alive, and insofar as it was ordered in some way was thought to be intelligent, and insofar as it took up space was thought to be made of matter. So they weren't in principle inclined to view anything as machine-like. They viewed things as much more organismically, and therefore there's a scene, for instance, in Plato's Republic, where Socrates says, I think that you can actually exercise the organ of thought the way people exercise when they do gymnastics. And this was a stream of the Western intellectual life. But it got submerged with the rise of modern science. Modern science has many potential beginning points, but one of them, of course, is Galileo. Galileo looked at the same planets that were thought to be animated by the Romans, so animated that they were given names like Mars and Jupiter and Venus, names of the gods, and he basically was able to determine that they were inanimate and they were moved about by forces. And his achievements were so grand that scientists began to think of the entire universe as a cosmic clock. And then biologists tended to take that model of mechanistic understanding and apply it to the organs inside our bodies. And William Harvey, who discovered that the heart was that the heart circulated the blood through vessels with valves, and it was essentially a pump, began mechanistic biology brilliantly. But of course, a pump is a machine. Shortly after, René Descartes, the philosopher, described the nervous system very much like a pump, and that there were currents that moved up and down the nerves. They were kind of an animal spirit or fluid, which we later learned were electric currents. And it soon seemed to scientists, whenever they were studying the brain, that the only explanations that were of interest were ones that saw it as machine-like. And even our modern medication models are very much a kind of lock and key model. They're very simple and mechanistic, and they surely simplify what's happening in the brain. Under the influence of Descartes, doctors began working with people who had had strokes, and Paul Broca had a patient who lost his ability to speak. The last words he said before his stroke, the last syllable was tan, tan. And after this man, who came to be called Tan Tan, died, he did an autopsy on him. And he found that there had been severe damage in this part of his brain, the left frontal lobe, and he got some other cases and soon declared that we speak with our left hemisphere. This was the beginning of what was called localizationism, and again, it merged into the idea that each part had just but one function. The problem was there were exceptions to this from the very beginning, but it was so appealing to think that there was one part and one mental function, and they always worked together, because the brain is so complicated. It's the most complicated object in the universe, that we really wanted to divide and conquer it to understand it. And localizationism took off and had many great accomplishments. But it went so far, it went too far. And the third reason that we miss the fact that our brains are plastic is because even when people did get better from strokes, and it happens from time to time, it was explained away. And what we would say is that this person's brain wasn't really damaged. It was just in shock. There was too much swelling. There was a metabolic derangement. And when they got better, all that happened is it just showed what they always had. They never lost cells. And I want to tell you one story about it, because it's the story of one of the first neuroplasticians I saw, Dr. Paul Bachirita, and his remarkable, remarkable family. And I don't have time to go into it, but he was the man who developed sensory substitution and showed that one sense can substitute for another, and that he was the person who actually taught people who had been congenitally blind since birth to begin seeing by getting input through their skin and tongues. Paul Bachirita was a polymath and a polyglot, the real thing. His father, Pedro, was a Catalan poet. And when Pedro was in his late 60s, he had a stroke. And he got the usual 6 weeks of rehabilitation. And then the doctors called, and they basically said to George, the other son of Pedro, come pick your father up. You have to find some place to house him for the rest of his life. And George was just a medical school at the time in Mexico. And he had no preconceived notions of the brain being fixed, because he hadn't taken that lesson yet. And he knew his father's indomitable will. So he took him down to the villa where they lived with a bunch of medical students, and he winged it. And he said, Papa, you learn to walk by learning to crawl like a baby, so we're going to get you to do that. And Pedro, he cooperated and got down on all fours. And when he was able to crawl, he took him outside of the villa where they were, and he got him to put one hand up on the wall, incrementally giving him some exercise, and got him to walk around. And then the neighbors thought it was very unseemly as to what they were making the old man do. And eventually he got up on all fours, and then he taught himself to type with the same sort of incremental principle, raising a hand, letting it fall, then letting just the wrist fall, then one finger at a time until he could type again, and eventually taught himself to speak. And 7 years later, he was remarried. He was a widower. He remarried. He went back to teaching, and he was climbing a mountain in Bogota, Colombia, when he had a heart attack, a fatal heart attack, and he passed away. And this is where the story gets interesting. The body was sent back to San Francisco, where Paul was working as a doctor, and he ordered an autopsy on his father. And a couple of days later, he got a call from the pathologist, and he went down to the lab, and there on the table before him were spread slices of his father's brain. And he looked at them, and he was numbed, but he also saw something there that was very, very important. When he looked at the tracks and the cells that governed movement in the brain, he saw that 97% of those cells and tracks had been wiped out. So what that meant was, this was not a case of a man who didn't have real cell death and just got the pump primed but with a little bit of exercise. What it meant was that the work that Pedro had done with George had completely and utterly reorganized his brain. And Paul Bachirita went on to be really one of the first champions of his generation of late brain plasticity, and developed these sensory substitution devices, which are so very remarkable. Now, there are many, many different kinds of interventions that are available for people who've had brain traumas. But in general, what happens is when there's a brain trauma, just as when you hurt your ankle, you guard it. You try not to use it so that you don't do more harm. But more often than not, that guarding causes difficulties. And one of the people I visited was Dr. Edward Taub. And what he was able to show was that after we have a stroke, the brain goes into this kind of shock, and you try to use your affected arm, and it just won't work. So you learn it won't work, and you start to use your better arm. And one of the core plastic principles we've learned is that it's a use it or lose it brain, and plasticity is competitive. There's a constant war of nerves going on inside each of your heads till the day you die for cortical real estate. And so the things that you do create coalitions of neurons that are very, very effective and work well together, and they're faster than the other coalitions of neurons. And they kind of bully out the other possibilities. So what Taub did with Michael Bernstein, a surgeon who in his 50s had a stroke, was he took his good arm and he put it into a sling so he couldn't use it, and then incrementally trained that. And that model works for many, many kinds of neuroplastic interventions. But what I want to emphasize to you is the competitive nature of plasticity. And this brings me to really the fourth reason that we missed the existence of brain plasticity. And that's plasticity itself. Plasticity is the culprit that causes us to miss plasticity. Because plasticity gives rise both to many of our flexible but also our most rigid behaviors. And I don't like to use metaphors in general about this because I saw how the machine metaphor caused a lot of difficulty. But one way to think about it is that plasticity is like snow on a hill in winter. And because that snow is plastic or pliable, you can take many paths down that hill. That's the flexibility. Not an infinite number of paths, because there's going to be rocks and trees in different places. And so they're going to constrain you somewhat. But if you go down that path and you like it, being human, you'll tend to favor a path very close to it or closer to it than any other path the second and third and fourth time down. And as you do that, you're likely to develop tracks in the snow. And those tracks will soon become ruts. And they'll become ruts, and you'll get tracks precisely because the plasticity makes for a snow that's pliable and flexible. And I believe that one of the things that we've all done is we have extrapolated, we've looked at our own rigid behaviors in life and the rigid behaviors of our peers and extrapolated many of these back onto the human brain. And we've done it not just as individuals, we've also done it as a species, because plasticity has this dual nature. So that's one of the most important things I learned in all my studies, what I call the plastic paradox. But I also learned that the brain, the resilient brain, is also a vulnerable brain. And one of the most interesting things is you can start, I said that there's something about culture here that's important. Just as children who have learning disabilities can be cured and their perceptual processors can go from having very low capacities for memory to becoming very sophisticated and efficient, so too can other parts of our perceptual apparatus. We now have incredible studies that show that people from China and related cultures wire up their brains very differently than people from Western cultures. This has nothing to do with genetics. This has to do with their experiences. But thought changes structure. And in culture, these thoughts change structure, millions and millions of people at a time. So when you show a still life to both, for instance, Americans or the British or Australians, and then you show them to people from Korea or China or Japan, quite unconsciously, quite involuntarily, they perceive them quite differently. And when you start testing them, you'll find that people from China are very, very good at spotting relationships between objects, including what we would consider minor relationships, and people from but they do badly on tests about the main point. And that people from the West have the opposite. They will find sort of the main object and be very good at it, but not great at understanding some of the relationships. And I believe these have had major impacts on philosophy and on politics and culture. And it's one of the reasons why when you're talking about issues of immigration, you have to start thinking of immigration or culture shock as a form of brain shock. But we also know that over a period of time, if a person moves from one of these cultures to the other one, or one of these civilizations really to the other, or back, their brains eventually rewire themselves. So plasticity turns out to be a very unexpected and very welcome discovery. And it'll be of use to humanity because it allows us to, in some ways, develop our brains in ways we didn't understand, and also to understand a whole number of diseases and difficulties that human beings have struggled with, because plasticity also can give rise to great rigidities. I thank you.
Thank you, Dr. Norman Doidge. Lots of questions, I'm sure, that you'll all have. And one of the ones on the tip of my tongue is, we've always assumed that the developing young brain is actually the most plastic organ. But my question, of course, will be, well, how do we extend that plasticity, optimise that plasticity in adulthood? And I was also interested to read that you think romantic love is one of the great examples of brain plasticity known to humans. I thought that was delicious as well. I'm really delighted to have two other speakers here tonight. One of Australia's true leaders in neuroscience in Australia, but also worldwide, and also following him will be a young gun at the beginning of her career in neuroscience. Can I welcome to the stage Frederick Mendelsohn? He's the director of the Florey Institute and R. Douglas Wright Professor of Experimental Physiology and Medicine at the University of Melbourne. Fred is the scientific director of the newly created Florey Neuroscience Institute, a really exciting, whopping initiative here in Victoria. He held a personal chair in medicine at the University of Melbourne until 1996 and was a senior physician at the Austin and Repat Medical Centre, and was also chairman of the Angiotensin-Gordon Conference USA in 1998, member of the Wills Committee on Health and Medical Research Strategic Review, was the 2001 Eccles Lecturer to the Australian Neuroscience Society. Countless other accolades to a huge figure in Australian science, and also past president of the Australian Neuroscience Society. Let's give him a warm welcome.
Thank you very much, Natasha. As you've heard from Dr. Doidge, the brain is probably the most complicated object in the universe that we know of. It's certainly one of the most fascinating, and I'm sure it's your interest in the brain that's brought you out on this cold Melbourne night. Neuroscience is really one of the most exciting branches of science. It's exciting because we've all wanted to know how our brains function since time immemorial, and how they malfunction in various disorders, but we've lacked the tools and techniques to study it. Now we have a number of enabling technologies to study the brain in great detail in many different ways. These include the Human Genome Project, which has given us an insight into the chemistry of the brain that was never before possible. Advanced new imaging, that we can look not only at the structure of the living brain, but which parts of the brain are active during various states. The discovery of stem cells in the brain that I'll tell you about in a moment, growth factors, and many others. So the next few decades are going to see extraordinary developments in understanding of the brain. You've just heard some of these extraordinary discoveries coming out, and there'll be many more. And there'll be major advances in diagnosing, treating and preventing brain disorders. Your brain is a marvellous thing. Within your skulls you have 100 billion nerve cells. That's approximately 20 times the number of people on Earth. And each nerve cell makes something like 10,000 connections with other nerve cells through specialised contacts called synapses. And at these synapses, nerve cells release specific transmitter molecules called chemical neurotransmitters. The pathways traced out by these connections encode our behaviour, our motor skills, our memories, everything that makes us human and that makes you a particular individual as defined by your genetic background and all of your experience. So even if you're an identical twin, you have a unique brain determined by the influence of your environment, your experiences, on top of your genetic pathways. As you've heard from Dr. Doidge, we used to think these pathways were fixed after early childhood. When I went to medical school, like Dr. Doidge, it was believed that the adult brain was incapable of repairing itself. But now we know that both of these beliefs are wrong. Pathways are defined by the strength of the synapses change in a dynamic way. While I'm talking, you're making and breaking synaptic connections at something like 1 million per second. The more you use a particular pathway, the stronger the synaptic contacts become. And I really like that analogy of skiing down the mountain and repeating pathways. The more you use the pathway, the better it gets, like a muscle that's exercised. And we used to believe that this was only a childhood event, but this capacity persists throughout life. There's no age at which you can't learn a new skill, that you can't learn a language, you can't learn, in fact, to modify your own thoughts. And you've heard a very dramatic way how sculpting these pathways can be enormously important in recovery from a stroke, say. This plasticity of the brain means that the capability of rehabilitation to improve recovery, to repair damage or impairment, has probably been greatly underestimated in the past, as you've heard. There's a second type of plasticity in the brain, quite apart from these pathways, these synaptic contacts. And it's a fairly recent discovery, and it's also amazing in its capabilities. This is the discovery of stem cells, neural stem cells in the adult brain. When I mention the word stem cells, many of you will think of human embryonic stem cells, the ethical controversy about whether we should or shouldn't try and use these to treat conditions like diabetes or spinal cord injury. But it certainly may come as a surprise to you to know that in your own brains there are neural stem cells which are capable of generating new nerve cells, new supporting cells, this process of generating new nerve cells is called neurogenesis. We thought it didn't go on in the adult brain. We thought maybe it persisted into early childhood, but then stopped. About 5 years ago, some scientists from the Walter and Eliza Hall Institute here in Melbourne and at the Howard Florey Institute were able to isolate some of these stem cells from adult brain, in this case a rat brain. We could culture them in a petri dish, and we could study what makes them divide, what makes them turn into nerve cells. If they turn into nerve cells, what type of nerve cells? And a range of molecules called growth factors have been discovered which regulate the division of these cells and what they become. This knowledge of stem cells and what regulates them means that an ideal treatment might be, instead of putting foreign stem cells from some other source into the brain, to use a brain's own capacity to repair itself. And this knowledge will have a huge impact in medicine. Christopher Reeve sadly died before his dream of repairing a spinal cord came to reality. We can't achieve this dream yet, but we understand the steps to get there. And all of the steps are feasible. they are, how to regulate endogenous stem cells, how to control what they turn into, and how to regulate their connections. And finally, I just want to mention that there's a surprising relationship between this process of neurogenesis, the stem cells producing new nerve cells, and mood and memory. Depression is associated with reduced neurogenesis, surprisingly, and antidepressants boost neurogenesis through a growth factor called brain-derived growth factor, BDNF. It's interesting that exercise also boosts neurogenesis in the brain, and exercise is antidepressant. So my message is, your brains are amazing objects. They're capable of extraordinary tasks, almost certainly much more than you'd ever imagined. And our capacity to recover from brain injury or impairment is probably much greater than we previously thought possible. Thank you.
Bit of neurogenesis just happened to me as I walked up those stairs, so I'll be up and down a few times. There's a few more nerve cells sprouting up in all the spots that I need them to be in. Thank you, Fred Mendelsohn, much appreciated. I'd like to introduce a new researcher at the Howard Florey Institute. She's at the beginning of her career. Emma is and we're very honoured to have her here because of this. She's at the pointy end of her PhD. She's in third year, working with Associate Professor Tony Hannon in what's called the Neuroplasticity Laboratory at the Florey. And I have to say, that lab is doing some fantastically interesting work. They're revealing that environmental enrichment, the sorts of experiences that we have in our lives can actually affect and change fundamentally the structure of our brain. And the other thing that they've revealed is that it can actually prevent the onset of serious diseases, specifically Huntington's disease, perhaps schizophrenia and dementia. Dementia can benefit also from that sort of external enrichment. It's very interesting work. And Emma's quite an entrepreneur too. I gather she was the co-owner and director of Brainiac, a student-formed company providing a 3D brain model called NeuroSlice for educating people about various diseases and disorders of the brain. And I'd like to warmly welcome her to the stage.
Thank you. So I grew up in the very beginnings of the couch potato era, where Sega Mega Drives and Nintendos were the in thing and constructing bush forts was going out of fashion. Now the couch potato has escalated. We have Nintendo Wiis that act as your personal trainer, allowing you to exercise without ever leaving the lounge room. Our fridges buy our groceries, and our cars drive us places. We can even live virtual lives and have virtual possessions and pets and friends on web-based programs such as Second Life. We are removing a lot of clutter from our lives, but what are we doing to our brains? The phrase use it or lose it is commonly used in our vocabulary. Most people associate this with the physical, exercise regularly, or lose your fitness. Though the same applies to the brain. And just like you need to vary the types of exercise you do, your brain also benefits from a diverse experience. As Natasha mentioned, I work with Dr. Tony Hannon at the Florey Neuroscience Institute, and in our laboratory, we are testing how exposing mice to stimulating or enriched environments improves their memory and mood and causes changes in their brain. So enriched mice are housed in much larger cages with toys to interact with ladders to climb, ropes to hang off, and running wheels to run in. We also remove them from their already large cage once a day and place them in a huge chamber with many different levels and 3D complexity. It's a bit like a mouse playground. I work with mice that are poor learners. I test their learning and memory using mazes that rely on a mouse's ability to use cues around the room to remember the quickest path to the finish, just like you would use the Eureka Towers to orientate yourself around South Bank, if you were Slip of the tongue there. South Bank, if you're new to Melbourne. So going back to the mice now, mice that use these cues very well get quicker and quicker at performing these mazes. And so what I do is I compare mice living in enriched environments to those living in non-stimulating ones, without toys and with much smaller space. While mice living in non-stimulating environments struggle in the maze, the enriched mice perform well. They get better every day. So after living in a stimulating environment, mice that are poor learners perform just as well as mice with normal learning ability. An environment with constant novelty and challenges improves their ability to learn. So enrichment has also been shown to stop the memory decline in ageing mice, and is also beneficial for a number of mouse models of brain disease. My supervisor, Dr. Hannon, while working in Oxford, looked into the effects of environment on Huntington's disease. Huntington's is a devastating brain disease that develops late in adulthood in those that have a mutation in the Huntington gene. Having this mutation means that people will get the disease, their life expectancy will be reduced, and they will experience debilitating motor symptoms and dementia progressively that get worse. And sadly, at the moment, there is no cure or treatment for this disease. Excuse me one moment. So before we knew that the adult brain is plastic, many thought there was little hope to combat dementia and brain disease later in life. Using transgenic mice with the Huntington mutation, Dr. Hannon housed half in an enriched environment and the other half in a non-stimulating one. He showed that the Huntington's mice that were housed in the enriched environment developed symptoms much later than mice living in the non-stimulating one. He could show that even though all mice had a mutation in the Huntington gene, not all of them got the disease at the same point. The environment slowed the onset. So we still need to know a lot more about Huntington's and how enrichment affects this disease. And since Dr. Hannon's study, researchers are starting to look into environmental factors that affect dementia and Alzheimer's and Huntington. So what does enrichment do to the brain? Housing mice in enriched environments encourages new brain cells to form. It strengthens the connections between these brain cells and also increases the expression of key molecules that provide support to the brain. So living in a stimulating environment drives the brain's plasticity. We think that these changes in the brain due to a stimulating environment may make the brain more resilient in the case of brain diseases. These changes provide a cognitive reserve, a bit like keeping your bank account in credit. We see examples of this in Alzheimer's disease and other forms of dementia, where there is evidence to show that high levels of mental and physical activity are associated with a reduced rate in cognitive decline. In fact, university professors are less likely to get Alzheimer's disease. So, what does all this enrichment mean for us, these studies on enrichment? Since studies have been published, Japanese architects have built apartments designed to challenge and stimulate the brain. The floors are uneven, the light switches randomly control a different light. You need to duck to get into certain rooms. The power points are high up close to the ceiling, so you need to devise methods to connect your appliances. And there's colour everywhere. Personally, I think living in an apartment like this sounds a bit stressful. But the concept of challenging yourself and working on training your brain on areas you're not particularly good at is one I personally live by. So as we have heard today, we know that the brain is incredibly plastic, and it also grows in response to our environment. And from enrichment studies, we can see that the environment we choose to live in affects the brain and our brain function now, but most importantly, affects our brain function later on in life. So that's food for the brain. Thank you.